Good morning, everybody. Thank you for coming to the third annual Synapse Roundtable. My name is Michael Graglia. I am the Managing Director of the Syngap Research Fund, and it is such a thrill to welcome our incredible speakers here today. In the interest of time, I'm going to hand this over to my colleague, Charlene Song-Rigsby, and let her introduce herself, and then our first speaker, Dr. Terry Joe. Charlene?
Great, thanks, Mike. Good morning. I am Charlene Song-Rigsby, and I'm the president and co-founder of the STXBP1 Foundation, and thrilled that we are having this roundtable this morning. I would like to introduce our first speaker, Terry Jo Bichelle. She is the executive director of Combined Brain, and she is going to be speaking on cures happen faster when unique disorders find strength in collaboration. Thank you, Terry Jo.
Thank you so much for having me. I really appreciate this. This is great. And I'd like to share my screen. All right, it worked. So far, so good. And now, can you see my slides okay?
Yes.
So, okay. So as Charlene said, I'm the founder director of Combined Brain. Combined Brain is a consortium made up of, at this moment, 26 separate patient advocacy foundation organizations, which you could see represented here behind my head and also on this slide. I founded this because I'd been working in the Angelman field for quite some time, for about 20 years. I have a son with Angelman syndrome. And what I found out in the past, that things have been changing in the past 20 years, quite a bit. So 20 years ago, when he was originally diagnosed, it was really rare to get a single gene diagnosis for a neurodevelopmental disorder. We didn't really know how to collect natural history. We didn't really know how to, we didn't have an ICD-10 code for our disorder. We didn't know the morbidity and mortality rates for Angelman syndrome. We really had to dive in deep and find out everything we possibly could about Angelman syndrome. And in doing this, what we found, and there are a few other neurodevelopmental disorders that were also early diagnosed, Prader-Willi syndrome, also on the same 15th chromosome area, Duke 15, also the same 15th chromosome. A few other disorders that were diagnosed really in the early days in the 90s. And we, at that point, really were working hard to show how unique we were, to show that we weren't actually, we shouldn't be lumped in with autism or lumped in with intellectual disability, that Angelman syndrome was really special because, in fact, it had all these unique characteristics. And so now that 20 years have gone by and whole genome sequencing is happening, and we are finding more and more and more disorders that we can identify with a genetic mutation every single day. Now things have changed a little bit. And so now what we found out is we've got to do this fine line. We've got to work really hard to find out what is unique about each disorder, but we've also got to figure out what we can share, what can overlap, what can we possibly do that somebody else has already done, so we don't have to reinvent the wheel. And what I've been telling people lately is, you're special, but you're not that special. And what we really need to do is work hard together and make sure that we can help each other get to our cures faster. So that's why I founded Combined Brain. Let me see if this will advance. Yes, it will. And it's a consortium led by patient advocacy foundations, because face it, even now, even with amazing clinicians, researchers in the field, really what we all need to acknowledge is that parents more than anybody else, are the leaders for rare disease research. Parents are the people who care the most about a disorder. Parents are the ones who can actually get the big picture of what life is like to live with a child with a disorder or a family member with a disorder. And so, in fact, these days, it's parents and patient advocates who are really leading the research for rare diseases. Of course, working tightly with the clinicians, the industry members, and researchers who are working on these disorders. But I mean, patient advocates are really key. So we are a patient advocacy foundation, and our mission is to fast track cures for all of these disorders that are part of combined brain. And we have 3 represented on the roundtable today. Phelan-McDermott syndrome, which is represented by Cure Shank, and STXBP1 disorder, and Syngamp mutation. Those 3 are 3 of the neurodevelopmental disorders that are part of combined brain. And what combined brain, our focus is on severe, rare, genetic neurodevelopmental disorders. The reason why we focus on that, there was a lot of work being done in autism. There was a lot of work being done in epilepsy. There wasn't very much work being done in cognition. And our disorders are all disorders that have severe cognitive disability. So this is why we are working together on looking for biomarkers and outcome measures for neurodevelopmental disorders. So when you think about, and I'm not going to read this slide, y'all have had plenty of time to read it while I've been talking. So if you think about cognitive disorders and treatments for cognitive disorders, I haven't had enough coffee yet, evidently. These are our members, current members. We do have a long waiting list that's building every day. And part of the reason for that is because there are new neurodevelopmental disorders diagnosed every day. So I do think that our membership is going to expand in 2022, but this is the current membership. Anyway, when you think about how, forgot to tell you, these are the 3 disorders that are here represented today. So when you think about a neurodevelopmental disorder and how are you going to measure, how are you going to do a clinical trial? How are you going to cure it? How are you going to figure out if a treatment is working? What we did in the past, and we did in Angelman syndrome, we made a mistake, which was we put the rodent models in the center of the picture. So we did a lot of great research in the lab. And when my son was diagnosed, there were no clinical trials whatsoever for human beings. But there were clinical trials going on in the lab for rodent models. And we were all focused on those rodents and focused on those rodent models and what we could do to improve their lives. And so what happened was that we did find some great treatments for rodents with Angelman syndrome. We reversed the phenotype in rodents with Angelman syndrome many times. And when we did this and then moved those compounds or those repurposed drugs into clinical trials with human beings, our trials all failed. So what we realized is that we need to put human beings in the center of this picture. And when I say we, I don't mean just the people I'm friends with or even the Angelman community or even the rare disease community. The FDA realized that trials were failing, and not just for neurodevelopmental disorders, all kinds of trials were failing because patients were not put in the center of the equation. And so the FDA in 2017 and 2018 introduced some guidances which really try to explain to industry and researchers that you need to put patients in the process from the beginning. You have to find out, what is it like to live with the disorder? What is your day-to-day life like? What would matter to you as a patient or to a family to change if you found a treatment? So we have all these amazing genetically related treatments now coming down the pike, so many kinds. Obviously gene therapy, but also antisense oligos, siRNA, miRNA, CRISPR. We have so many possibilities for genetically related treatments that could actually really correct the underlying disorder beneath any of these severe neurodevelopmental disorders. And if it worked, if one of these therapies worked and it changed something, it improved something, what would matter to patients to improve? What could we measure that would show that quality of life is better for that patient and that family? So now what we do is we've got to actually find out what matters to patients first. It's one of these things that's so obvious, it just drives you nuts that people didn't do it before. And then we have to collect the natural history on that. So we have to ask questions about life that matters to patients on a regular basis over time, not just once right before a study, but over the length of a person's lifetime. Then we have to figure out which of those things can be measured in the models, the rodent models, even the cell models. And then we have to figure out what endpoints can we develop for trials based on what we can measure in the humans and the rodents and what matters. And then we do the clinical trials. That's how we get clinical trials to succeed. So this is what Combined Brain was built to do. And by working with all the member organizations, we really help all these patient advocacy organizations identify that they themselves have the most responsibility and the most access to all of these parts of drug development that are represented above this timeline from, the gene ID to a cure before the FDA. So it's really at this point up to patient advocacy foundations to lead the charge for natural history. And really, this might be a little controversial, I hope not, but the patient advocacy foundations really need to own the data for natural history trials, because researchers come and go. Y'all get new jobs, you retire, you decide to move to another institution. Researchers come and go. Industry comes and goes. Clinicians come and go, but the Patient Advocacy Foundation, they are going to care about this disorder until it's cured, and then after it's cured, probably to help find patients and get them to the treatment they need. So natural history data, it's got to be spearheaded or coordinated, or at least owned, accessed by the Patient Advocacy Foundation. Plus, they're the ones who have the lists of patients. They're the ones who are going to recruit all the patients to get in the natural history and then to get in the clinical trials. And actually, if we do natural history right, natural history can be a control. This is the holy grail of natural history. We all got to work hard on that. And then disease concept studies and disease burden studies. These are really important in drug development. you have to understand what is the burden of a disease on a family or a patient. How, you know, if a patient has a severe neurodevelopmental disorder, what has to happen in the whole family? Does somebody have to give up work? How many medical appointments are there? How much money does it cost? That's disease burden. And then disease concept, I'm going to talk about in a minute, is what the disease is like in daily life. And then it's really up to patients to give input to patient-centered outcomes. How are you going to measure if a treatment's working without asking a patient? So all those things are on the top of the timeline. And then everything below the timeline, that's got to be done really spearheaded by researchers, by industry, by clinicians, but they're going to need stuff from up on top of the timeline. So to get biomarkers, you're going to need biosamples. So you've got to coordinate for those. And same thing with everything else on the bottom of the timeline. So as part of our 2021 initiatives, we worked hard to get the foundational work done for all of these various components of drug development. And because time is short, I'm not going to go into details on any of these, but I hope that you can get a hold of me through the website combinedbrain.org, or you can email me. And I'm just going to talk about a couple things right now that we've been working on that are really pertinent. One is a biorepository. How are you going to get all those biosamples? I mean, how are you going to get those biomarkers without biosamples? And this is where I tell people you're special, but you're not that special, because it's a lot easier to work across disorders to figure out biomarkers. What proteomics can we do? What are there possibly byproducts that could go that are in blood that could go across a whole bunch of different disorders? Could you measure not just what's specific for the disorder, like gene expression, but also what might go across disorders like neurodegenerative products? So we need biosamples. So we've opened a biorepository that is open to any neurodevelopmental disorder, and researchers can access these samples at really the lowest cost you could possibly get by with in this field. We also do these conceptual models of disease. We do the draft of the conceptual model. And today we have on this call two disorders that have gone forward to do the fully the final disease concept study. So in a nutshell, what a disease concept study is a literature review looking at every symptom ever reported. And then, I see Mike popping up, so I better hurry up. And we ask every question that you can think of about what your daily life is like. And we add all of those symptoms to a big disease concept map. And from that, we draw all of our goals and objectives. So one of them showed that of our original 20 disorders, communication, especially expressive communication, was one of the #1 issues for every single disorder. And so we have a new initiative working with Duke to expand a communication measure called the ORCA, the Observer Reported Communication Measure, to 12 different combined brain disorders. Amongst those, the very first 3 are Syngap 1, STXBP1, and well, Phelan-McDermott is the 4th. And the reason why those are our first few disorders to be looked at is because when we did our disease concept models, we could compare the disorders to each other to see who was similar and who was different. And as the ORCA was actually invented or developed for Angelman syndrome, we wanted to start out with the disorders that were closest to Angelman at the beginning, Syngap 1 and STXBP1, and then Phelan-McDermott. And so now I'm going to conclude my talk with asking if you have any questions. And did I leave time for a single question? Oh, you're silent.
Not really, but thank you so much. I think one of the reasons we asked you to kick this off is because we're so proud of being a part of Combined Brain, and the whole theme of this roundtable is collaboration. So this is a great opportunity for us to show our communities and our colleagues that we are working with you and all these great organizations on the screen to do anything we can to help our kids in the most efficient manner possible, because our kids are sick, as you rightly said, and we care deeply. In the interest of time and out of respect for all the other speakers, I'm going to plow right through. Thank you so much, Dr. Bichelle. I will see you soon at the FAST meeting. I think, and over to Dr. Dahlman, who is a neurologist, a neuroscientist, I should say, at the University of Miami. And like all of our speakers today, Dr. Dahlman is working on a couple of our genes. So in the interest of time, I will let you take it away. Thank you so much for being here.
Thank you so much for inviting me to participate in this roundtable. And I was really, I'm glad I'm following Terry Jo. I use animal models, but we're trying to do this work in the context of also being cognizant of natural history. And so I hope to tell you a little bit about our integrated approach. We've started with Syngap 1 and Shank 3 models of zebrafish models. Okay, so here's a quick rundown of what I'm going to go through. I'm going to give you a brief introduction, talk about zebrafish models, talk about integrating the clinical and basic studies, and then give a little summary and future direction snapshot. So when, as someone who works in an animal model and not even a rodent model, a zebrafish model, we focus on symptoms that we feel like we can make mechanistic insights on, and where the mechanistic insights might inform treatment strategies. And so the two that we've focused on are altered sensory processing and gastrointestinal distress, both I'd say symptoms that we know a little less about. And then one of the things that I've learned in my foray into this field is that the gut itself is an organ that functions through sensing information, nutrients in the lumen, and producing a behavior, peristaltic movements to aid in digestion. So this idea of sensory processing being an important symptom that has big impact on quality of life, this altered sensory processing could play out in the gut as well. And as all of you know, the speed of identification of genetic forms of neurodevelopmental disorders has been very rapid. And so this is just a slide from Satterstrom et al. 2020 that shows some of the single genes of large effect that have been linked to neurodevelopmental symptoms. And I'm circling Shank 3 and Syngap. I think I've put in the year in which variants in those genes were linked to neurological conditions in humans. And you can see that Syngap was linked much more recently than Shank 3. And as such, the natural history that's known for different conditions is very different, just in part based on when that linkage occurred. Okay, so my lab focuses on these sensory motor symptoms. And in particular, we've taken a deep dive into GI symptoms, in part because so little is known about how those symptoms arise. And actually, in attending Felon McDermott Family Foundation meeting and talking to families, these GI symptoms were symptoms of big concern. And so that's how we got into looking at these. And so this circle here are molecularly defined forms of ASD with documented GI distress. And this is an incomplete list. before agreeing to, or not, before the meeting this morning, I looked at STX-BP1, and there are also GI symptoms described in that condition, even though it's not listed on this set. Of these genetic forms of neurodevelopmental disorders, 6 have, in animal models, of 6 of these forms have shown that there's reduced motility, reduced movement in the GI tract. And that movement is critical to digestion and gut function, because it's part of a reflex that when the gut senses nutrients, it induces these contractions, it induces mucus secretion and hormone secretion that aids in digestion, coordinates with the brain, coordinates behavior. So one of the hypotheses that we're interested in testing is that this reduced motility is a common aspect of ASD-linked GI distress. And then you can see that 3 of these animal models are zebrafish models. I'll show you why zebrafish are useful for looking at GI movement. But there are a lot of genetic models in zebrafish that haven't been examined for GI motility. So zebrafish are a good model for this because by 6 days after fertilization, they have these relatively translucent larvae that are eating on their own. And you can actually watch, this is a movie of a larva that's been fed, and the food includes 6-micron beads. So you can actually watch these peristaltic, the part of the reflex that I was talking about, in vivo. And that makes it quite straightforward to quantify what's going on. Different from us, right, where digestion is happening inside and it's not visible. In addition, we've been looking at the response of the nervous system to external stimuli like light. And as I said, we're starting to think about this sensory motor integration, both at levels of gut and brain. Okay, so what have we found? So this is, again, these peristaltic movements that are response to feeding in wild type. This is our Shank 3 model. And believe it or not, the movie is going. So there's a little twitch that happens here every now and then. And this is a fed Syngap model larva. You can see that there's actually quite a bit of motility in the gut, but it's not coordinated from a anterior to posterior direction. And so we can quantify this behavior, and we can show that, so for instance, in wild type, there's a particular rhythm that is also seen in Syngap, but is slower in Shank 3. And so this echoes this need to kind of understand, these studies suggest that, so yes, both are showing GI symptoms, both genes seem linked to these GI symptoms, but manifest in slightly different ways that are important to understand. So the other test that we do in the zebrafish is whole gut transit. And what happens when you, in both Shank 3 and Syngap larvae, so what's happening here is we fed them beads in wild type. And in wild type, what happens is the beads are consumed and then make their anterior posterior journey through the GI tract to be expelled. And this usually happens within 12 hours of consumption, the first expulsion. Whereas in Shank 3, at this intermediate time, the beads all accumulate at this junction between the intestinal bulb, which is like the stomach, and the upper intestine. And only by 24 hours do you get expulsion. This is true in Syngap as well, and this is quantified here. So what are the cells that mediate this GI reflex? Well, it turns out that your gut is lined, it's sprinkled with these sensory endocrine cells called enteroendocrine cells in light blue. And those actually make most of the serotonin in your body in response to either mechanical or chemical stimulation in the lumen of the GI tract. Those are activated and talk to enteric neurons that activate the smooth muscle cells. So this circuit is really important for taking food in the lumen and inducing the reflex that is mucus secretion and contractility in the gut. And to go deeper into this circuit, we've been collaborating with Liu Huaye and John Rawls at Duke University. The other thing that points us to these enteroendocrine cells as being important is that when we quantify enteric neurons and enteroendocrine cells, which are important for motility, both Syngap and Shank3 mutants have reduced numbers of enteroendocrine cells, which could mean that they're less sensitive to luminal stimuli and therefore don't produce the normal response. And I think these, both from the therapeutic perspective and from animal model studies, most of the attention with these genes that are linked to neurodevelopmental disorders has been focused on the central nervous system. But the gut also has a significant nervous system through the enteric neurons. And, in the collaboration with John Rawls, he does this single cell sequencing of the intestine. And here is, so what they do is they take the intestinal cells and do RNA sequencing on the single cells. And what's shown here are the different types of intestinal cells. And then on this axis are genes that have been linked to these severe neurodevelopmental disorders and where these genes are expressed in the intestine. And these first 5 rows are the enteroendocrine cells. So you can see that Shank3, catnap, orexin, CDKL5, SCN are all enriched in these sensory endocrine cells, indicating that they could be really functionally important to the GI distress that's impacting these individuals. You can see that other genes linked to ASD are more generally expressed throughout the intestine, and Syngap is not expressed in the intestine of the zebrafish, but is expressed in the enteric neurons. And so this raises the possibility that therapies will have to be cognizant of those cells in the gut that will also need to address the genetics of those cells. Okay, in addition to gut sensory motor integration, we also conducted studies of responses to visual stimuli in the Shank, in our Shank model. And what we find, so this is an assay that is done in zebrafish where you have light to dark transitions, and anytime you turn off the lights, there's a lot more swimming, and that response is dampened in both in 2 different models of Shank3 where the mutations fall either in the N terminus or the C terminus. And what you can see if you map the responses in the brain, the responses to that turning off the lights are that the pineal gland turns on and the hindbrain and spinal cord turn on, corresponding to this increased movement. Whereas in the Shank models, the pineal turns on, but it fails to activate this hindbrain region, which is associated with the movement. So this suggests that the sensory processing deficit is in the hindbrain. So to test that possibility, we transplanted cells from a wild type animal into a Shank3 mutant animal. Here you can see at 1 day post-fertilization, these cells have populated the hindbrain, and this is at 5 days, or actually 6 days when we do the behavioral assays. Now these Shank3 model has a wild type hindbrain, and that actually rescues their response to the light stimuli. So that this shows that the hindbrain is actually, Shank3 in the hindbrain is really crucial, at least in the zebrafish, for responses to light stimuli. Okay, I know I'm losing time, so I wanted to tell you that we're also trying to contribute to better understanding GI symptoms in neurodevelopmental disorders. And one way is, as mentioned by Terry, it's really important to have objective measures. And so it turns out that you can measure whole gut transit time by eating a muffin with a blue dye in it. And this was actually done on a epidemiological scale in England, but not with kids with neurodevelopmental disorders. And we also have, in collaboration with Baharak Moshiri, who's at Atrium Health, we developed a blue muffin that copies the tests that are used that involve X-ray and to measure whole gut transit, with the idea that if we could use the blue dye and look for arrival of a blue poop, that would provide whole gut transit that could be done at home and without intervention. And this is
ready, Sissy?
We have seen whether this is a child with a Syngamp mutation, and she's willing to eat
Michael,
it. And
do you
here's
want
another
some more?
child who's
You want
being
more?
offered a blue muffin.
Do you like that? Yeah, it's edible, huh?
Okay, so we've also developed an app to try to do, in a prospective manner, gather these symptoms related to GI distress. And what we're hoping to do is use these measures in patient populations at the same time as we're looking for mechanisms of GI phenotypes in zebrafish models to develop strategies for these GI symptoms more generally. And this is the wonderful group that has supported this research. It's been a huge team effort. And so I think I have a teeny amount of time for questions if anyone has them.
Dr. Dahlman, do you see the question in the Q&A panel?
Yeah, I do. And I think it is possible that increasing, so there are, you can, yes, this is something that we're planning to test. It's likely that it would work in the model that has problems with the enteroendocrine cells, but we have to test that.
Hi, everybody. I'm sorry, I've lost my voice. I'm Geraldine Bliss. I'm the president and co-founder of Cure Shank and the mom of a 23-year-old son with Phelan-McDermid syndrome. And I'm delighted to introduce you guys to Dr. Kolovzan. He's a professor of pediatrics and psychiatry at Mount Sinai School of Medicine. He is the clinical director of the Seaver Autism Center and the PI of the Phelan-McDermid Syndrome Natural History Study in the Developmental Synaptopathies Consortium. Alex has a tremendous amount of experience in characterizing kind of the natural history of Phelan-McDermid syndrome and also in conducting clinical trials in Phelan-McDermid syndrome and other neurodevelopmental disorders. So Alex, thank you for joining us today.
Thank you, Caroline. It's very nice to be here. I'm so sorry that you don't feel well. That looks and sounds painful. Maybe you need a blue muffin. This looked awesome. I was really glad I got to see that. All right, I'm going to share my slides. All right, can you guys see now my full screen?
Yes.
Okay, cool. All right, I'm sorry that I missed the previous whole talk, but it sounded like we were talking about sort of translational work from animal models to clinical trials in kids. That's also where our work started. I'm going to focus today mostly on the kids and just talk a little bit about what we've learned in terms of the natural history and the phenotype of Phelan-McDermid syndrome, some of the challenges we face, and then a few of the ways that we're trying to address those challenges. So this may be redundant at this point, but Phelan-McDermid syndrome is due to Shank3 haploinsufficiency. These are small deletions or mutations on the terminal end of chromosome 22. We know Shank3 acts as a master scaffolding protein in glutamatergic synapses. We know it induces neuronal formation. It represents a potential target for treatment. These kids all have profound global developmental delays, a whole range of dysmorphic features, none of which are specific. They all have language impairment. They all have some degree of intellectual disability. About 80% probably have, or 86% in this case, have low muscle tone. A lot of that is also related to GI dysfunction, as you heard about, probably the most common of which is constipation. Sleep disturbance is a really pronounced problem that affects kids profoundly, and also, of course, parents. And I guess this is an epilepsy group. So about 25% of these kids will have epilepsy. There's probably no specific kind of epilepsy or seizure phenotype. It's really all over the map. And probably up to 40% of kids, if not more, will have underlying EEG abnormalities without specific clinical correlates. And many will also just have non-specific histories of febrile seizures. So that's Phelan-McDermid syndrome. In 2013, Geraldine approached me and said, you need to put in a grant to study the natural history. I said, no way, we're not ready. She kept at it and paired us up as a Phelan-McDermid syndrome consortium with tuberous sclerosis and P10 or Cowden syndrome. And so we formed this developmental synaptopathies consortium all around these synaptic genes. And our goal was really to comprehensively characterize the phenotype in children, adolescents, and now adults we've included, using a whole array of medical, genetic, behavioral, and cognitive measures. We really set out to add to the literature by doing prospective assessments to following kids over time, and to really sort of ensuring that what we understood about the phenotype, at least to date, was correct. We were very interested in tracking the natural history. Obviously, that's critical for developing clinical trials. We don't want to be picking symptom targets that are highly variable. And we want to be thinking about what are potentially tractable in the context of treatment. We were very interested in developing biomarkers, first using neuroimaging and now focusing more on electrophysiology, and I'll talk about that today also. And then the real main driver here is to develop new treatments. So we want to eventually start to assess safety, tolerability, and the feasibility of experimental therapeutics. We've done that in 4 or 5 different clinical trials, relatively small scale, all at a single site. And we're really looking now to kind of expand to leverage our whole network. So the network exists across now 5 sites focused on Phelan-McDermid syndrome. There's probably 10 sites in the DSC overall. But Mount Sinai, Rush, with Latha Soria and Elizabeth Berry-Kravis, Boston Children's with Mustafa Sahin and Sid Srivastava, the NIH, the intramural program with Audrey Thurm, and Stanford with John Bernstein. We have collected data for about 140, 150 patients at this point, 90 in our first round, and we've now been funded for a second round. And we're doing baseline assessments, and we're following people out after 1, 2, 3, and now 4 years as well. So it's a really robust longitudinal study at this point. The second iteration, which was just funded, I think, 2 years ago, is now including kids that are younger than 18 months, or rather younger than 3 and older than 21. So we're really trying to capture the entire lifespan here. Well, one of the first things we've done, and we continue to add to it and refine it, is develop a kind of a standardized assessment battery. And we're really interested in a whole range of features from cognitive ability to adaptive behavior, language, motor functioning. One of the things that kind of unifies us is this autism phenotype, even if only about 60% of kids with Phelan-McDermid syndrome actually meet criteria. So we do a lot of digging into the autism phenotype, especially now around sensory symptoms. And then other kinds of behaviors, sleep, regression has become a very, very important area now to focus on, quality of life, of course, and any kind of longer term cognitive changes that might occur. So this is our battery. We recently published a study looking at the relationship between the phenotype and the genotype, probably the largest, at least prospectively collected study to date. And I thought I'd just put some of the features up that we now kind of established the prevalence where, as I said, hypotonia is probably the most common feature, gastrointestinal dysfunction, most commonly constipation, apraxia, ataxia, spine abnormality. So there's a whole kind of, broad and highly variable phenotype that affect people from, from 86, 90% of people, all the way down to 4 or 5%. I think dysmorphic features gets a lot of attention because it certainly alerts people to the concern and it triggers the need for genetic testing. But there's really no specific, I would say, dysmorphic features. Probably one of the more common ones and more unique ones to feel determined are dysplastic nails. But again, nothing would be considered in any way pathognomonic. I guess I'll make a point also, I haven't seen it in the chat yet, but I'm sure Geraldine will ping me to do this. As it turns out, a lot of the epilepsy panels, so when you have a kind of a seizure phenotype and you order genetic testing, and there are a number of different sequencing panels that don't include Shank3. So while chromosomal microarrays will pick up a large number of kids because of copy number variants affecting terminal 22q, you really need to do sequencing to pick up obviously smaller point mutations and sequence variants. So it's just important to be thoughtful about which panels you're ordering because Shank3 is obviously one of the most common causes now of autism and neurodevelopmental disorders, even if not specific to epilepsy. So it's critical to think about exactly which genes are included in your panels. Other features include things like disrupted sleep, as I mentioned, anxiety. More and more we're understanding now that kids at around puberty are really vulnerable to developing pretty pronounced neuropsychiatric decompensations with the emergence of symptoms that are consistent with bipolar disorder and even catatonia. It doesn't happen in all the kids, but the prevalence seems to be becoming greater and greater as we learn more. We're becoming more attuned to it. And then this issue of regression, although regression is characteristic of neurodevelopmental disorders and autism broadly, up to about a third of kids, it's certainly very characteristic also of a subset of people with Phelan-McDermid syndrome. And some of the things that I start here, which I wasn't going to cover very specifically, but there seems to be a relationship between the size of the deletion, where smaller deletions and point mutations are actually people that are at higher risk of regression. It may very well be because even at baseline, they have higher levels of language and higher levels of adaptive functioning perhaps, but they're more prone, of course, to also regressions. In terms of cognition and adaptive behavior, these are kids that are severely to profoundly affected for the most part. Obviously, there are people on sort of both ends of the spectrum, but for the most part, we definitely see this as a pretty severe intellectual disability. And we have a bunch of different language tests, including more objective tests like the EVT and the PPVT, again, all of which just show pretty pronounced impairment, where only about half these kids are really verbal in any meaningful way. More than half are considered to be minimally verbal. And some of the importance of these measures is that they all represent possible tools to be using in the context of clinical trials. So to sum up some of the natural history, and I didn't present what the phenotype looks like over time, although when we've looked at it, it does seem to be stable to a large extent, with the exception of some standardized measures where we're seeing some growth into deficit just based on the comparison to typical peers. But in general, we're seeing that most of these measures seem to be relatively stable, and many of the tools that we're using are potential tools that we could be using in clinical trials because of their stability and just the test retest over time. So the natural history will clarify the phenotype for us, and I think we've done a lot to really better understand that and establish the prevalence of various features. I think we've done a lot to try to identify targets for therapeutic interventions, and I'll talk about some of the tools that we're using. Eventually, we hope to be able to identify demographic, genetic, environmental variables that actually correlate with disease outcomes, the ability to try to predict who's at higher risk of regression, the ability to try to predict who's at higher, who has sort of more likelihood of developing meaningful language. All these things are really critical in terms of just helping families and helping understand what the course of illness will be. And then we're also focused now on developing better consensus guidelines to establish both assessments, monitoring schedules, and then of course treatment, at least to the extent that we know about what works, And so we have a big working group now that's been developed across many, many different specialty areas, and hope to put out a consensus guideline sometime in the next 6 months. Okay, so that's the phenotype. And then in terms of the clinical trial readiness, this is really where we're all kind of so challenged, right? Because I think if we had the best medicine in the world, or even a gene therapy, perhaps, we wouldn't necessarily feel convinced that the tools that we have to measure change over time are especially sensitive or relevant specifically to Phelan-McDermid syndrome. So generally speaking, you think about clinical outcome assessments as either patient-reported, caregiver-reported, clinician-reported. You can combine instruments to create composite instruments, and then the ideal, of course, are like more objective tests. So in Phelan-McDermid syndrome, we don't have patient-reported outcomes, but we're still really kind of committed to trying to figure out, how to assess treatment benefit, right? And according to the FDA, treatment benefit is really determined by how a patient survives, feels, or functions as a result of treatment. And there are so many different symptom domains that really profoundly affects these kids and affect their quality of life. So we're really thinking a lot about across virtually all these domains, whether it's caregiver, clinician, or objective tests, to try to target specific domains and develop much better refined tools that are actually valid specifically to Phelan-McDermid syndrome. So one of the first things that we did was actually just took existing tools, right? So again, this idea of an autism phenotype is important to us. It's one of the things that unifies the Developmental Synaptopathies Consortium across different genetic conditions. And the SRS is a very common tool. It's used in autism and neurodevelopmental disorders. It's one of the most commonly used one. It's also used in some clinical trials as an outcome measure. So we were interested in how it performed in Phelan-McDermid syndrome because we had a suspicion that it was not going to be especially relevant. And in fact, it really seems to have limited utility in general in populations with severe intellectual disability, including Phelan-McDermid syndrome. One of the challenges that we saw was that the majority of respondents endorsed really the greatest severity of autism. So there was a really pronounced ceiling effect. This is despite the fact that less than 60% of the kids met criteria for autism. We found that there was no significant difference between the mean SRS scores, whether you had autism or didn't have autism. So that was obviously a problem in terms of this measure. I think in 2017, Sturm developed a 16-item questionnaire that was a sort of a briefer version of the SRS, but it was supposed to be for people with with intellectual disability, but still had kind of limited performance in our Phelan-McDermid syndrome population. So this was kind of what we predicted. It was a good sort of first pass in terms of assessing existing tools, and it was disappointing, although we learned a lot. And I think I've since kind of abandoned this particular measure in Phelan-McDermid syndrome. Another domain, in addition to autism and sort of as part of autism, really is sensory sensitivity. And more just based on our clinical experience, it seems that there's a relatively unique sensory profile within Phelan-McDermid syndrome, at least in comparison to idiopathic autism. So one of the other things that we did is just took the existing short sensory profile and did a comparison between Phelan-McDermid syndrome and idiopathic autism. And overall, we found that Phelan-McDermid syndrome kids actually showed less overall sensory sensitivity. And here, higher scores are actually more typical performance on this measure, as opposed to most measures where higher scores are worse. But in particular, we saw that there was a kind of a specific subset of the kids who had more sort of low energy or weak symptoms, or more kind of in general hypo-reactive. And that seems to be pretty specific to Phelan-McDermid syndrome. Well, not specific necessarily, but at least compared to autism more broadly, these kids have a lot more hyposensitivity in general. So some of this also led our group, and Paige Seiper in particular, who's our chief psychologist, to develop a specific sensory assessment for neurodevelopmental disorders. So this is really the first assessment that's combining a clinician administered observation with a caregiver interview, and it's capturing sensory sensitivity. So if you think about the ADOS, in many ways this is mimicking the ADOS play, but tapping specifically into visual, tactile, and auditory sensitivities. It's appropriate for sure for use in severely affected and minimally verbal populations. Kids with autism show significantly more sensory reactivity symptoms on the sand compared to typically developing kids, so we know that. And then again, when you look at Phelan-McDermid syndrome in comparison to idiopathic autism, we see that these kids are much more prone to hypo-reactivity symptoms compared to idiopathic autism. So this is thought to be now a beneficial tool for both research and potentially for clinical use. It's now been licensed, and there's going to be ways to kind of get trained up on it and use it clinically as well. We have a method of trying to test all of our measures, and both existing measures and measures that we're trying to develop in the context of clinical trials. So we will always be doing sort of pre and post assessments in the context of treatment. So one of the things that we did with our IGF-1 trials was we did the SAND before and after treatment, and we found that actually kids with Phelan-McDermid syndrome showed both significant improvement in sensory symptoms following 12 weeks of treatment, but in particular, that improvement overall was driven by improvement in the hyporeactivity domain. So that was kind of an important finding for us, and has now led to a number of grants that have really embedded this tool in a much more consistent way. Just looking at the time. Oh, I have 5 minutes. Okay, maybe I'll skip this one. So the other types of measures that we're trying to develop use electrophysiology, both auditory event-related potentials, but also visual evoked potentials. So VEPs in particular are a non-invasive technique to evaluate the functional integrity of visual pathways. They're recorded from the occipital scalp in response to a contrast reversing checkerboard. The major, and let's see if you can see my, oops, sorry. You can see my mouse, right? So the major peaks, this N75 peak reflects glutamatergic activity, and the P100 reflects the kind of subsequent inhibitory response. So the balance between excitation and inhibition is reflected in this waveform. This is a very typical waveform of virtually anybody that's typically developing, and it can be an infant or an adult. But in Phelan-McDermid syndrome, what you see is sort of a massive attenuation of this N75 peak, and then of course also an attenuation of the reflexive inhibitory peak. That's exactly what you'd predict based on knowing that the Shank3 gene is an anchoring protein for glutamatergic synapses, right? And so when you compare people with typically developing folks to people with idiopathic autism, to Phelan-McDermid syndrome, and even to Phelan-McDermid syndrome siblings, what you see is a dramatically reduced N75 peak. And so we think about that as a potential marker to hopefully predict treatment response, and the goal is to kind of match that up with clinical outcomes, specifically around the sensory domain. You can do VEPs pre and post treatment with drugs that we know act as kind of growth factors like IGF-1. And here in particular, the results show that improvement in the active treatment group, this is a very small subset, so this is very preliminary data, but improvements are seen in the beta and the low gamma frequency bands. So the thought is that this is potentially an important measure, and it definitely reflects the fact that there's increased excitation as a result of treatment with IGF-1. I think biomarkers are, everyone talks about biomarkers, and they're ideal because they're objective, but they're only really going to be, I think, extremely useful if they're tied tightly to sensory or clinical symptoms in general. And so here, this shows that the kids with Phelan-McDermid syndrome show more visual hyporeactivity compared to idiopathic autism in typically developing kids, and that the extent to which that hyperactivity occurs is actually pretty tightly correlated with the magnitude, or the amplitude rather, of the excitatory peaks. So we do think there's a nice relationship between sensory reactivity, the VEPs, and we're showing also relationships between the SAND here, which is our sensory measure, and the VEP response, particularly around the glutamatergic peaks. So this just gives you kind of a little bit of a sampling of the kind of work that we're doing, the way that we're thinking about clinical trial readiness. There's many other groups, and we have a lot of other tools that we're testing. But the hope is that once really viable treatments are available to test, we'll be ready with the right kinds of measures, and we'll certainly know enough about the phenotype and the natural history. So just to summarize, so we obviously want to be carefully phenotyping these kids, and that's, I think, what we've been doing. We've got a number of target symptoms of interest that we think have really important impacts on quality of life and clearly reflect the phenotype. Establish the natural history is an ongoing project, and we're starting to learn more and more. We want to refine existing clinical outcome assessments, and we want to develop some new tools. And of course, we have to develop and validate biomarkers, both as sort of a stratification tool but also hopefully as a way to predict treatment response. And this is our team. This is way back when we were able to actually meet together in real life. I guess I'm happy to take any questions. I got it exactly in 20 minutes.
You nailed it. I don't know if we have a ton of time for questions, unfortunately, but I'm so grateful for Geraldine for inviting you to this, because I know Charlene and I are looking at this salivating and, thinking about when we'll be able to see such rich data, and even, dreaming about being a part of the DSC for STXBP1 and Syngap. So thank you so much. I have questions, but in the interest of time and out of respect for the next speaker, who'll be talking about Shank 3 and Syngap 1, I'm going to keep this going. So much, Dr. Kolovzan. Our next speaker is Dr. Connie Smith-Hicks from Johns Hopkins, the Kennedy Krieger Institute. I can't say enough nice things about her, but in the interest of time, I will just let her wow you herself. Dr. Smith-Hicks, take it away, and thank you for being here. And I will brag that she is on our SAB. We have the privilege of her. Over to you, Connie.
Okay. Hi, everyone. Can you see my slides?
Yep.
Okay, wonderful. All right. So thanks, Mike, and the organizers for inviting me. So I'm going to talk about sleep and synaptopathies. I'm going to briefly just talk about an overview of synaptopathies, and then why we chose to study sleep, with some focus on some of our work looking at sleep in Phelan-McDermott and Syngap 1. Then I'll end with a little summary and some next steps, and perhaps some thoughts about limitations of the work that we've done so far. So, when we think of synaptopathies, we're thinking about disorders of the nervous system in general, but it can certainly be a consequence of dysfunction at the synapse, both peripherally or centrally. But for the purposes of this talk, I'm really going to be thinking about what occurs centrally, meaning in the central nervous system. So there's been a significant increase in the number of genes that have been linked to NDD, and currently they're thought to be over 2,000 genes that have been associated with neurodevelopmental disabilities. Many of them function at the level of the synapse, both presynaptic and postsynaptic. But the schematic here features 3 genes that are being discussed today. So STXBP1, Shank, and Syngap 1. And what you'll notice is that STXBP1 primarily functions in the presynaptic compartment, whereas Shank and Syngap primarily function in the postsynaptic compartment. And Shank and Syngap are involved in synaptic plasticity mechanisms, whereas STXBP1 is primarily involved in release of neurotransmitters. So we know a lot about the clinical features of all 3 disorders. Just in general, we know that they are certainly associated with a complex clinical phenotype that will include global developmental delay, intellectual disability, autism, and epilepsy. And we know that sleep is a common feature in many neurodevelopmental disabilities. In individuals with Syngap 1, it's reported to occur in about 62% of individuals, and then in individuals with Phelan-McDermott, in anywhere between 25 and 50% of individuals. And I think from the previous speaker slide, I think we had probably 62%, if I recall, or 56% if I recall. Now, although sleep difficulties are reported in patients with intractable epilepsy, and we see significant epilepsy in individuals with STXBP1, the prevalence of sleep disturbance in that population is not entirely clear. So the focus of our conversation is really going to be related to Shank and Syngap 1. So why study sleep? Well, it certainly is an area of unmet need. And we know that sleep is critical, and good quality sleep is critical for many physiologic functions. It supports neurogenesis, plasticity, brain development, and behavioral function. And any parent of a child will tell you that poor sleep negatively impacts behavior, and there's this bidirectional association between sleep and mood, and certainly sleep and cognition. Thus, poor sleep not only impacts the child, but also impacts the caregiver. Sleep is not just a common problem in neurodevelopmental disabilities, but it is pretty prevalent in the general pediatric population, and approximately 25%, so 1 in 4 children who are otherwise typically developed have sleep problems. And depending on the nature of the neurodevelopmental disabilities, the prevalence of sleep can be anywhere between 50 and 95%. Sleep difficulties may manifest either with difficulty with sleep onset, sleep maintenance, problems with having increased daytime sleepiness, or breathing challenges occurring during sleep. So work from several groups in preclinical models of Shank shows that the Shank protein is involved in circadian rhythm of, and these studies are actually done in rodents, but the synaptic level of Shank protein varies over the course of the day. And the changes correlate with changes in serum melatonin. Now, mice lacking exon 21 in Shank 3 are shown to have less sleep during the dark period. And this disruption in Shank 3 expression is also correlated with disruption in expression of many of the genes that regulate the circadian rhythm. So based on this, we know that Shank 3 is important for the regulation of sleep. And just a note I want to make, the exon 21 in Shank 3 is actually one of the hotspots for many of the pathogenic variants that are seen in individuals with PMD. Like Shank 3, Syngap 1 is also an important regulator of sleep. And Syngap 1 is expressed in the suprachiasmatic nucleus, which is the brain circadian clock. Its levels vary over the course of a day. And in rodent models that are haploinsufficient for Syngap 1 have increased activity during the dark phase relative to wild-type mice. So why did we compare sleep in Syngap 1 and Phelan-McDermott? Well, we opted to look at these two disorders because, one, they have shared clinical features, and their genes converge on shared molecular pathways and have shared physiologic consequences. We studied sleep in this population, and the patients were recruited from 3 sites, 2 in the US and 1 in the UK. We recruited patients with Syngap 1, Phelan-McDermott, and typical developed siblings. We took all comers from as young as 3 years old on up. What we noted was that there was no significant difference in the number of males versus females for either of the 3 groups that were included in this study. I shared earlier that sleep is a common problem in both Syngap 1 and Phelan-McDermott, and so it's not surprising that many of the patients were taking at least one medication that is routinely used to treat sleep. You can see that melatonin is by far the most common medication that's used in both groups of patients. We used the children's sleep habit questionnaire to evaluate the sleep profile. This is a parent-reported screening measure, and it evaluates behavior or sleep difficulties that are both behaviorally mediated as well as medically mediated. The CSHQ has been validated in kids between age 4 and 10, but it's been used in children younger and children older. And a total score that is greater than or equal to 41 suggests clinical significance of disrupted sleep. Not only do we look at the total score, but we are also able to parse sleep features along 8 subscales, looking at certainly resistance to going to sleep, delayed sleep onset, how long the child remains asleep, anxiety, parasomnias, and breathing-related difficulties, as well as daytime sleepiness. So using the CSHQ, we first asked, is there a difference in the total score? And then the subscale scores between all 3 groups. And we show that individuals with Syngap 1 and individuals with Phelan-McDermott both had more severe sleep problems when compared to their typical developed siblings. However, individuals with Syngap 1 had more sleep difficulties than those with Phelan-McDermott, and that's indicated by the higher total mean score. This difference between Phelan-McDermott and Syngap 1 seems to be driven primarily by differences both in bedtime resistance and a difference in daytime sleepiness, where you see that Syngap 1 individuals had higher scores on both. We also noted that nighttime awakenings, parasomnias, sleep disordered breathing, and sleep duration were common problems in both Syngap 1 and Phelan-McDermott individuals. Because there is, in general, thought to be an age difference in the manifestation of sleep difficulties, we divided our cohorts in those who were younger than age 11 and those who were older than age 11. And another motivation for doing that is, again, because the CSHQ was validated in those younger than age 11. So we asked, is sleep difficulties in these disorders age dependent? And so we looked at this pattern, and we can see again that individuals with Syngap 1 had a higher total sleep score, so a worse sleep profile than individuals with Phelan-McDermott. And we saw that pattern in those who were younger than 11 years of age, so those who were prepubescent, and that's consistent with what we saw when we looked at the larger group. We also see that the difference is the difference between these 2 groups is driven in this case by sleep anxiety and also by daytime sleepiness, and a difference in parasomnias between individuals with Syngap 1 and siblings as compared to Phelan-McDermott and siblings there was no difference between those with Syngap 1 and those with PMD, and both disorders had more increased severity of parasomnias as compared to typical developed siblings. We then ask, well, what is the profile of sleep differences that we see in the older groups? We can appreciate immediately that there was severity of sleep challenges in both Syngap 1 and Phelan-McDermott when compared to typical developed siblings in many of the sleep subscales, and that there was no significant differences between older individuals with Syngap 1 and PMD. So in summary, we see that sleep is a common problem in NDDs in general. Both the Shank3 and the Syngap 1 protein are important regulators of sleep. We see from our data that there is greater sleep difficulties in individuals with Syngap 1 when we compare them to those with Phelan-McDermott, and we believe that this is dependent on age. So the younger Syngap kids have more sleep difficulties compared to younger individuals with Shank3, whereas the older individuals with PMD seem to have more sleep difficulties than their the younger individuals. Parasomnias, nighttime awakening, and disordered breathing are common features. I think, this data suggests that both the total and subscale scores in the CSHQ can be useful measure for evaluating sleep. I think that it'll be important for us to look at the utility of this in natural history study and what it tells us about the sleep profile over time for populations with Syngap 1 or other synaptopathies. One of the questions we often wonder about is, how does the CHSQ or any other measure that we use, how does that change over time when we think about treatments? And that's something that we are looking into. I mean, we generally put many of our patients on medications that treat sleep. And so this would be an easy thing to do, at least in the clinics. So I want to stop here and acknowledge certainly the families and children of Syngap 1 and Shank3, the parent advocacy foundations. The work was done in collaboration with Jimmy's group in Texas and Andy's group in the University of Edinburgh, and certainly all our funding organizations. So I think I have probably 2 minutes for questions. Okay, and I see a question here in the Q&A that says, do you see a wearable tracker as being a way to follow sleep habits over time? Absolutely. That's actually one thing that we are embarking on, and we're in the process of analyzing that data. I think wearable tracker, we may be able to correlate activity and delayed sleep onset or latency to falling asleep with the CSHQ. Some of the other measures, I think that might be challenging, but yeah, that is something that we and others are doing, and we just don't have the data yet to share. Okay. And do you think that sleep could improve with treatments? So absolutely. Genetic treatment. So do you think that sleep could improve with genetic treatments? Absolutely. I think, if we are, there are many approaches that are being proposed for genetic treatments, whether it's antisense oligonucleotides, or other modalities, whether it's read-through or many other approaches. And if we are able to get target parts of the brain that certainly are involved in regulating sleep, then it would be my expectation that sleep, we should be able to improve sleep. And sleep generally, in my opinion at least, is a low-hanging fruit. It's one of the things that we are able to treat to some degree in many of our patients. I wonder, in the data that we showed, you might have noticed that the sleep onset delay was, there was a lower score, subscale score, although significant when compared to typical developed peers. But I wonder if that lower subscale score and consequently less impairment in delayed sleep onset is related to the fact that many of these kids are on medications that their role is to increase sleep, so target delayed sleep onset. And so I wonder if we are being effective in shortening the latency to falling asleep in many of the patients with the current medications we use. My expectation is that targeting sleep will improve behaviors. Will it be a cure? I don't think so, but it'll be one of the treatment interventions that we would likely employ in as we think about how we treat patients with Syngap1, Shank3, STVX, STXBP1, and other similar disorders.
Thank you so much, Dr. Smith-Hicks. Sleep, I'm glad to hear you say low-hanging fruit. I hope that will be the case. It's obviously something that can really impact quality of life. I am delighted to introduce Dr. Chung. She is a clinical and molecular geneticist at Columbia University. She's one of the world's experts in the genetic basis of human diseases. She's also the director of clinical research at Simon's Foundation Autism Research Initiative. She's the PI of SPARK and Simon's Searchlight, which both aim to understand genetic neurodevelopmental conditions. Thank you so much for joining us, Dr. Chung.
Thanks, Geraldine. I'm really thrilled to be here. Let me see if I can share my screen the right way. And hopefully you can see everything. So I'm very pleased to be here, and I'm going to show you hopefully some data that I hope many people can use, and always looking for input on how we can do better. This is part of the umbrella program that I'm calling Simon's Searchlight, although as Geraldine said, we also sponsor SPARK, which is a study that I think of as feeding into Simon's Searchlight. So Simon's Searchlight is predicated on the fact that individuals with neurodevelopmental disorders are clearly not the same, and we can start to understand better, and we must in terms of getting to treatment, subdivide by genetic basis, but also realize that as we subdivide, we can see synergies, and obviously the synaptopathies are a great example of that. This is just showing you the current genes and copy number variants that we include within Simon's Searchlight to say that the knowledge is still increasing in terms of the genes that are involved. And so we continue to expand our genetic conditions that are included within that. And I'll also point out we continue to iterate in terms of inclusiveness. And one of the things we've been doing is increasing the number of languages that we can collect and distribute data in, both in terms of just online writings as well as the languages that our research team speaks. With this, the goal in terms of Simon's Searchlight is again to be able to make for participants, i.e. families, make it relatively easy to participate and make it scalable in terms of the distribution of the information so that we can all learn faster. And as we're doing this, I'll show you some dimensions. We've been thinking especially about how to get more resources than just data to researchers, and I'll get into some of the specifics of that, but I can really feel a shift in terms of the impact that's having on the field. As we're doing this, the low-hanging fruit, so to speak, are to be able to get online data. The reason we've done that, some of you know that as we started this out as something we called Simon's VIP at the time, it was more burdensome on families. We had them come in person, do a couple days in terms of evaluation. We still have those data freely available to researchers, but we realized that we couldn't include as many people due to travel restrictions, especially with COVID. The online community afforded us much more flexibility to understand in real time how COVID was impacting our community, as well as being able to continue data collection. So as we've done that, number one, we collect the data online as well as with genetic counselors who review and be able to really, I think, help us in terms of getting really clean, robust, valid data, having genetic test results from families that we can validate their diagnosis, sharing that information back with families and community groups, as well as making that freely available to researchers around the world. And as we're doing this, as I said, we hope that this is partially powering readiness for clinical trials, as well as being able to power the researchers who are going to develop those understandings of basic mechanism, as well as some of those treatments. This is just an example of the online measures that we have, and the medical history interviews that we do. The point, or one point to this, is that this is not one and done, but this is longitudinal, appreciating that things change over the life course, challenges change as people evolve, and so we continue to collect data as individuals are changing over that life course, both from a medical point of view as well as a developmental point of view. Again, I'll show it at the end, but all of these data freely available. As we're doing it, we want to make sure that this is as I said, available to researchers, but also that this is more short-term affecting clinical care and clinical guidelines. And one relatively easy way to do that we found is through gene reviews, being able to, that's for those of you who don't know, it's an online resource, again, open access, formatted in a consistent way where a busy practitioner can be able to log in for the gene of interest and find sort of clinical care, what they should be doing in terms of health surveillance or interventions. And again, something that we as genetic professionals continue to update. We take that as a serious responsibility. So that's something that we've powered many of our Searchlight genes to be able to enable gene reviews. I and my teams have done some of those reviews, but many others in the community have done that as well. And then also making sure the variant level data is in ClinVar. So again, it's without any identifiers in terms of participant identifiers, but making sure that diagnostic laboratories have free access to the information so that more individuals get correctly diagnosed as they're going through diagnostic testing. I will say all of this in the very careful curation of those genetic test results we found to be incredibly valuable, although about 90% of the individuals who come into the Searchlight portal, once we review their genetic test results, about 90% of them in fact have the right diagnosis, but there are a good 10% where that's not definitive based on the testing we've had so far. One of the other things Searchlight does is to actually pay on our dime for the genetic testing, for instance, of parents, if they have not been part of that genetic testing process, to make sure that we know whether this was an inherited or de novo variant. That also helps in terms of interpretation of that variant, as well as getting additional information on additional people that may be valuable. And again, that's value added in terms of what Searchlight can do. All of these data available, again, the URL is right here in terms of Safari Base. We have incredible data navigators to also be able to help researchers understand the data in addition to data release notes and data dictionaries. But this is regarding specific clinical data, as I said, genetic data, but also importantly biospecimens. So whether they're DNA samples, induced pluripotential lines, other types of cell lines, making sure that's available. And importantly, an opportunity that we have called Research Match. So this is an opportunity for us to be able to advertise to the community by gene or by groups of genes, research studies that are IRB approved through investigators around the world, to invite them to be part of their research, whether it's online, whether it's in person, but be able to match those researchers with individuals in those gene communities. And that's been, I think, is going to continue to be a very effective way to allow families to participate, not just in Searchlight, of course, but much farther. This has been an important resource that we've been creating, induced pluripotential potential stem cells. I'm listing here and showing some of the conditions that we've added recently in green, the new cell lines. These are all done again at the expense of the Simons Foundation and freely available both to basic science researchers in universities as well as in commercial settings. Within this, in some cases, we have coisogenic corrected lines so that I think they have both good controls as well as allelic spectrum so that we have many different variants within the same gene, so that if there are any allele specific either differences in mechanism or differences in severity, those can in fact be studied by researchers. So again, all of these freely available. We've had a call out to the community to vote democratically on what lines are necessary or helpful to the community, and always looking again for feedback if there are lines that we don't yet have established. Within this, I started to describe that this research match opportunity is something that's always ongoing and active. And so this is something, again, that we can take literally as studies are ready, whether these are clinical trials or survey-based studies or looking for clinical readiness or a particular measure, whether it be sleep or something else, to help you with your studies. And again, to be able to do it across conditions, which might be helpful in terms of looking across synaptopathies or synaptopathies versus other types of genes. Data sharing really is something we believe very, very strongly in. Again, we, just to be clear, this is all de-identified individuals. We try and make sure that they are in fact qualified researchers by requiring a research distribution agreement, which says that they agree not to try and identify individuals within these studies. And as we do this organically, it grows so that data, for instance, from research match studies organically comes back into Safari base, so participants don't have to keep answering the same questions over and over again, but can be able to do something once and have many, many people be able to benefit from that. As we're doing this, we do try and cooperate with other groups that are also collecting data. In particular, Citizen, for instance, has been focusing a lot on EHR data, being able to work together so that we can increase the power of all of the different data sources that are available. With this, as I was saying, one of the advantages perhaps compared to some registries that might be individually focused on a gene or a group of genes is there are important differences that at least I've noticed in comparing across our different conditions. Individuals, for instance, who may be wanting to explore a particular phenotype, it's very easy to be able to look across genes and be able to see what are the particular genetic conditions that might be associated with either very little of a particular symptom or a high frequency of that symptom. And that might be useful in terms of comparison across those to understand that biology or behavior better. In particular, for the synaptopathies, as we're talking about, we do have very, very rich data collections on some of these groups. I give a lot of credit to the family group leaders. It's in large part because of their efforts to rally the troops and to emphasize the importance of research to their communities. And I won't go through exactly all of the numbers of individuals as of today, but we do have large data sets for all of the ones that I've boxed and shown and might be of interest to the group here. In particular, and not surprisingly, all of them are associated with neurodevelopmental conditions, but in particular, if we look at some of the major sort of conditions that we see, everyone has some degree of cognitive neurodevelopmental issues. Most have issues with epilepsy or seizures, and autism or ASD on the left is seen in a large number, but certainly not everyone. And I want to be clear that although Safari, I think, is known for being an autism sort of umbrella in terms of the research, this is really a genetics first approach in which clearly not everyone within these genetic groups has autism, nor are we solely interested in autism as a manifestation. So in fact, we fund many researchers who study these conditions not solely for the purpose of autism, but for epilepsy or other behavioral issues. The types of things we give back to family are these research snapshots that I'm showing you here. We try and do this as regularly as we can, and especially as we have new data releases. So you can see, for instance, with STX-BP1 in the last data release we had of 68 individuals, or Syngap1 with 31 individuals, being able to see the types of things, the types of distributions of frequency of manifestations that we see. And again, we hope that this information is valuable to families and will continue to enrich it as we think about the numbers increasing, responses to certain medications or problems with certain medications, but deeper detail that we often hear families wanting to have answers to. Within this, we see many different manifestations manifestations beyond what I just said, some that are more observable directly by clinical observation, and so getting increasingly deep in terms of some of those assessments, but again, being able to see some things that are common features across many of the synaptopathies, and in fact, across many of the neurodevelopmental conditions that we study. So with that, the other thing that I'll say is that this last year, some of the investigators know we had a request for applications, or an RFA at Safari, that was specifically giving funding opportunities to investigators, not solely to use Simon Searchlight data, but to be able to go deep. In particular, it was genetically focused, so to go deep in terms of understanding mechanism, but importantly, driving us closer to therapeutics. And so I think we've just been able to finish the review of those RFA, or those grant submissions, and in fact awarded those applications out. And I'm very excited to see that we are, I think, going to be able to give significant amounts of dollars at the Simons Foundation. I think based on the success this last year, we're thinking this is going to be something to continue doing, to be able to catalyze on the therapeutic side, but specifically for the conditions that we have under Searchlight. So I hope, as a brief summary, this again is something in terms of what might be a resource available from the Simons Foundation. Again, the URL to be able to get any of the cell lines or any of the data that we have here. We have scientists on the Safari team that can help you in terms of, again, any taking requests for anything else that you'd like to see that we don't already have, as well as RFAs that we have throughout the year for funding for any of these programs. So I'll stop there, but thanks again for including me.
Thank you so much, Dr. Chung. It's a real honor to have you in this event. We have a couple minutes for questions. Like Dr. Smith-Hicks, you've actually left some time, which is amazing. Until questions roll in, I just want to say that SRF, I'm sure my colleagues at STXBP1 and Shank 3, we do encourage our patients to take part in Simons Searchlight. And I'm particularly thrilled to know that through partnership with your incredible team, Jennifer and others, we have also been working on getting a connection with Citizen, who's going to talk next. So hopefully we'll be able to get our community taking advantage of both of Citizen's medical records and of all of the incredible investment that Simons does. I just can't say enough. I'm so grateful and honored to have you here. If there are no questions about, wait, here they come.
So the first question, in terms of a significant reason why Syngap 1 has half the pool of patients in Safari base is the other genetic difficulty. We support everyone, so we're not filtering anyone. It's simply who volunteers. So if anyone would like to get the word out, certainly we encourage you to, and we continue to expand in terms of capacity based on how many families we have. So you keep them coming, and I promise we'll keep up with you. So I'll give you that challenge. And then, do we have resources for families as far as understanding their disorder, genetics, biology? The answer is yes, absolutely. So we support family meetings, both online, it's been during COVID times, but also in person. Oftentimes we'll have sessions like Genetics 101, or we've had sessions that I've done about gene and cell therapy and how to be able to understand that. So there are lots of resources, and we've recorded all of these. So they're enduring resources that can be available to families asynchronously anytime, and especially I did that because I know families are newly diagnosed every day, and we want them to get that information right away when they're in that state of panic and trying to learn as much as they can. So we can certainly follow up, but they're scattered in a few different places, but YouTube is an easy way to find a lot of the information content, and we're happy to share that as a resource afterwards.
And I will also say, Pat Breland, because you're one of ours, we are working to correct that. There are incredible opportunities from the Simons Foundation, like the cell lines. And I will also mention that the Simons Foundation supports Autism BrainNet, who SRF is a proud partner of. I know it's a horrible thing to think about, but we encourage our families to consider donating their loved ones' brains after they pass. Hopefully we don't have to face that for a while, but it's important for research. So Simons just does so much, and we want to see our patients in that. And hopefully by building this bridge with Citizen, who we'll hear from next, and Simons Searchlight, we'll be able to up those numbers and really take advantage of the generosity of that foundation.
Mike, just one more, since you mentioned Autism BrainNet, an unabashed advertisement, if any of you can get the word out to scientists. We will be having, or actually just released, a request for applications for scientists to use the resource from Autism BrainNet. So again, we're not just creating those resources, but we're giving funds to scientists to do the science. So please encourage them to apply. We'd love to have them use it for your groups.
Absolutely, thank you. With that, I will go from one of the giants in genetics and registries to somebody I humbly think is one of the future giants. One of my favorite SAB members, Ellie Brimble from Citizen, is just one of my favorite people. And I will let her talk about Citizen and thank her in advance for all of her good work for all of our kids. Go ahead, Ellie.
Oh, well, thank you for that very kind introduction. Let me get set up. Can everyone see the slides okay?
Yep.
Perfect. So thank you very kindly for the introduction and for inviting me to speak with you today. I'm going to be sharing some information on behalf of Citizen, which is now part of the larger Invitae family. And so to start off with an introduction to Citizen, this is a patient-mediated platform, so patient-centric, patient-focused, and it really allows participants to collect and store their medical records. And then what we do with those medical records once they've been collected is transform those into research-ready data sets. So the goal is to make these computable, digital, and relevant so that they can be shared at the user's discretion in order to support research and clinical programs. The FDA has recently come out with guidance around the collection of real-world data, and so we've worked to ensure that the way we approach data collection is consistent and of high quality. And one of the ways that we've sought to approach this is by taking a framework that includes two different levels. So our topmost level we refer to as the disease model, which essentially serves as the conceptual foundation for the data products on the Citizen platform. And so what the model is doing is it's defining in advance what types of data is extracted, ensuring that information is relevant and important to the conditions on the platform. And then it also establishes relationships between those different types of data. So rather than just having kind of a linear outline of various symptoms and procedures and interventions, we're able to create a network of information describing how things are connected to one another. Within the disease model, we have also built out a manually curated ontology. And the intention of the ontology is to be able to harmonize and normalize the information that's collected. So we can essentially think of this as a really large data dictionary, ensuring that each term or each piece of data that we collect from medical records is assigned a standard way to talk about it. So we all know that many individuals who have a rare diagnosis are followed by many different providers. And so what this allows is even if providers and doctors are talking about the same types of symptoms in different ways, we're able to normalize across that variation. We've been incredibly lucky to work with some fantastic organizations, including two that are represented here today. And so what I wanted to take an opportunity to do was share some of the data that we've collected in order to encourage collaboration and further use of this data. So by the end of the year, we expect to have complete data for 70 individuals with STXBP1 and 105 individuals with Syngap 1. In order to illustrate the range of data that we collect from longitudinal medical records, we've randomly selected 50 individuals from each cohort, and I'm going to be sharing some data with you for those individuals. To start with, here are some characteristics of the 100 participants spread across STXBP1 and Syngap 1. On the left-hand side here, we're looking at the distribution of males and females. And so we can see that it's a roughly equal distribution for both populations. And then these two graphs here are showing the distribution of age, whether that's age today or age at diagnosis. And so we did not see any significant difference between the two cohorts in terms of age today or at age of diagnosis. And we can see the averages here are relatively close to each other. One of the things we pride ourselves on at Citizen is our somewhat ruthless approach to collecting medical records. We do our best to ensure that we are fully capturing the patient's interaction with the healthcare system, whether that's at one institution or at multiple institutions. And so I wanted to share some information around the medical records that we collect to give you a sense of the breadth and scope of data. So comparing STX-BP1 to Syngap, when we look at the number of institutions that we've collected medical records from, for Syngap, this is about 6 institutions. Oh, my apologies. For STX-BP1, this is about 6 institutions. And for Syngap, it's about 5. But you can see that it ranges all the way up to 17 or 18. In terms of the number of individual documents, so these represent individual clinic notes, imaging reports, lab reports. For STX-BP1, we were looking at an average of 212 documents per patient, corresponding with about 750 pages. And for Syngap, it was slightly less, about 175 documents, corresponding with approximately 650 pages of information. But again, you can see that there is a wide range of volume of data, in many cases dependent on the presentation of the individual. So as I mentioned in our introduction slides, we have a disease model which defines different types of information that we collect. And one of the broader categories of data we refer to as clinical diagnosis, but it's really meant to capture any diagnoses, phenotypes, signs, or symptoms that a patient may experience. And so one of the exercises that we did to get a sense more broadly for the diagnoses of individuals with either STX-BP1 or Syngap is we took those unique diagnoses and mapped those back to a broader organ system. And this is one of the benefits of using a standard terminology, which allows that propagation in a somewhat automated fashion. And so what I'm showing you in the graph here, in the light green color, we see STX-BP1. And then in this tealy, bluey, greeny color, we see Syngap. And we're showing the number of unique diagnoses mapped to each organ system across those cohorts. So for example, looking at the neurology bars at the very top of the graph, we see that there were 408 unique diagnoses spread across the, or counted across the population compared to 327 in Syngap. And so unsurprisingly, we see that some of the more commonly affected organ systems or broader categories of systems are neurology, neuropsych, which we've included some behavioral diagnoses as well, musculoskeletal, GI, and eye. And so this is certainly consistent with what's been presented here today as well. We can also dive in. So, first we talked about diagnoses more broadly, but we can also narrow in to focus on more specific diagnoses. And so here I've pulled some diagnoses that are relevant to either one or the other or both of the conditions presented here today. And so we can look here at the frequency of epilepsy, any developmental and epileptic encephalopathy, any movement disorder phenotype, an autism diagnosis, or behavioral challenges. And so already here we can start to see some differences in the phenotypic spectrum of these two conditions. So in STX-BP1, we see that we're observing a higher frequency of developmental and epileptic encephalopathies, as well as movement disorder. Whereas looking at Syngap, there appears to be a higher frequency of autism diagnoses and behavioral challenges. So we can certainly do this across the full spectrum of diagnoses that are extracted to start to tease apart some unique differences between various conditions. One of the important approaches to data extraction that we take is every unique data point is assigned a date. And so this allows us to characterize the phenotype across an individual's lifespan. And so one way I wanted to highlight that today was to talk about the presenting diagnosis or symptom in both STXBP1 and Syngap1. And so when we look at STXBP1, we can see that there is kind of a very striking presentation where epilepsy is certainly the most common presenting feature, whereas comparing that to Syngap, we see a little bit more variability in what might be the presenting symptom. But we do see a spectrum here with some overlap, where epilepsy and seizures, delays, and hypotonia are common amongst the two conditions. Whereas Syngap, we see reflux as one of the more common presenting features, as well as feeding difficulty with plagiocephaly and respiratory distress in the STXBP1. We also looked to see whether there were any differences between the age at onset. So how old were individuals when they were first reported to have a diagnosis or symptom? And we do see that individuals with STXBP1 present significantly earlier than those with Syngap1, for an average of 5.2 months compared to 11.1 months. I also wanted to look to see whether there were any differences in the time to diagnosis. So what this graph is showing us is based on when the patient first presented, how many months did it then take for them to achieve their diagnosis of either STXBP1 or Syngap1? And so we didn't see any significant differences between the two, with the average time to diagnosis for an individual with Syngap at 60 months, and then the average time to diagnosis for an individual with STXBP1 was around 59 months. I wanted to get a sense of whether the specific presenting diagnosis impacted that duration. And so we broke the presenting features out into epilepsy versus all other diagnoses. And while we do see that it appears those with epilepsy did achieve diagnosis earlier than those where the presenting symptom was not epilepsy, this didn't achieve statistical significance. We can also dig into specific phenotypes. So we know for both Syngap1 and STXBP1 that epilepsy and seizures are an important clinical endpoint to characterize. And so looking at the age of epilepsy onset, we do see a difference in age of onset. So looking at the graph here at the top, we broke out age of epilepsy or seizure onset into neonatal, so presenting within the first month of life, infantile onset, presenting in the first year of life, and then childhood onset, which would be after the first year of life. And we can see that there is a striking difference between STXBP1 and Syngap, where the majority of individuals with STXBP1 have seizure onset within the first year of life, whereas this is really a minority of patients with Syngap1 and epilepsy, where we see that those individuals are presenting in childhood. And we can also see that the age of onset of epilepsy was statistically different between STXBP1 and Syngap, where again, we're seeing that individuals with STXBP1 have seizure onset much earlier in life. We also pull out information related to medication use and the duration of medication use. So here I'm showing an example specific to anti-seizure medications. And so we're looking here at the most commonly tried anti-seizure medications in STXBP1 and Syngap. And so we can see that there are some unsurprising names here. We have Keppra, phenobarbital, Onfi, Topamax, and ACTH in STXBP1. And then for Syngap, we see Keppra as the most common, followed by Onfi, Lamictal, Valproate, and ethosuximide. We also see that in both cohorts, people are trying a lot of different anti-seizure medications. So on average in STXBP1, we're seeing people are trying around 5 different anti-seizure medications ranging from 1 to 16. And in Syngap, this is about 6 different anti-seizure medications ranging from 1 to 18. We can also take a look at how long medications were used. So even though some medications may be tried more frequently, we wanted to try to get a sense of whether or not some medications were felt to be more effective than others. And one of the ways we wanted to try to approximate that was by looking at the duration the patient was on that medication. So in these graphs, we're looking at the duration of medication use normalized to the duration of epilepsy, and we see some differences here. So in STXBP1, we see a couple of new medications that were not necessarily the most commonly prescribed, but they did appear to be used for a longer duration compared to the full number of different seizure medications that were employed. And the same here with Syngap1. And one thing that's interesting with Syngap is, if you'll recall from the previous slide, Keppra or levetiracetam was the most commonly tried medication. But here we're seeing that it's not one of the medications that was used for the longest duration. So this is an interesting approach that we could take to try to understand whether or not certain medications or combinations of medications are felt to be effective by the treating team. And then for the last couple of slides, I wanted to talk about data access. One of the themes of this meeting today is collaboration, and so we certainly want to make sure that those interested in these conditions know how to access the data we're collecting. So all of the citizen data collected is done so under a broad consent, which has received a determination of exemption through Category 7. And so in order to access citizen data to support academic research, we simply require the following: a study protocol outlining the use of citizen data that has achieved a determination of exemption through Category 8, which we are happy to assist with, as well as a signed research agreement. And if you have any further questions, feel free to reach out to research@citizen.com and we'll be happy to support you. And then I did also want to just make a plug that we are working to connect patient data across other platforms through common identifiers, with Simon Searchlight being one of the examples where this has been very successful. And so really what we're trying to do is to prevent data from being siloed in different places. And so our goal is really to be able to connect data that lives across various platforms to create a really rich research data set that can be used to accelerate clinical programs in these conditions. So, one of the exciting things about now being part of the Invitae family is we're hoping to be able to add genetic data, whether that's the specific genetic variant, causative for a person's diagnosis, or whole genome sequencing, for example. We're now going to be able to layer that onto the longitudinal clinical data that we extract ex citizen, whether that's any patient-generated health data, biospecimens, and we have also successfully connected to individual patient registries as well. So this is part of a larger effort amongst many groups to ensure that data can be connected across various platforms where an individual patient may be participating in. And this is just a plug. If you're here at AES, we do have a poster with some more Syngap data. So feel free to come by to say hello. I'll be standing by the poster tomorrow. Thank you very much.
And Ellie, I think we have one question in the Q&A panel.
Oh, that's a great question. And I may recruit Dr. Chung to help answer this as well. I can speak for the citizen data. We are focusing primarily on extraction of medical record data. So taking the information that lives in the existing medical record from each time you visit a physician or provider and transforming that into a normalized data set. And we are working to be able to share data between the Simon Searchlight program as well. And if you'd like to add to that, Dr. Chung.
So as Ellie said, we're very complementary in terms of we're really family facing to be able to make sure we get that perspective and get very granular data in terms of these annual evaluations. So the two are very much complementary in terms of that. And so we've really striven to be able to make sure, as Ellie said, that we're not siloed and that you can put the pieces together to be able to get the most out of the combined data sets.
Great, thank you. Well, thank you so much, Ellie. I think it's just so exciting that we're really starting to build up data sets for these rare neurodevelopmental disorders. And as Ellie said, bringing together and integrating that data is critical for us to be able to extend our understanding and leverage the work that's happened to gather that data. So thank you. So I'll move on to introduce our last speaker for today, Dr. Ben Prosser from University of Pennsylvania. He is on the STXBP1 Foundation SAB, and he is also an STXBP1 dad. And so I will hand things over to Ben.
All right. Thank you, Charlene. Thank you to Mike and Charlene for inviting me to talk today. I'm thrilled to follow Ellie as well, who sort of set up my talk nicely in terms of presenting the clinical presentation. And I'm going to talk about our work trying to develop antisense oligonucleotide based therapies for STXBP1 and Syngap1 disorders. So these sort of how it started, how it's going memes are making their rounds on social media these days. And so this is mine. I started my lab at Penn about 7 years ago. And for almost the first 5 years, we studied exclusively the mechanics, the molecular biology of the heart. And this was going well, we thought. But then we shook things up. And these days, about half of my lab is studying the circuits of the brain and genetic therapies for neurodevelopmental disorders. This is very different than the heart, of course. It's not a great career move, I think, if you can avoid it. But I couldn't avoid it because in 2018, my amazing and beautiful daughter, Lucy, was born, and she started having seizures a few days later and was diagnosed with STXBP1 encephalopathy a few months after that. And so it was a tough first year for Lou. We couldn't get her seizures under control, but eventually we did. And now, as you can see here, Lou is a very happy kid, as long as you were paying full attention to her. But Lou is also 3 now, and she can't crawl or walk or talk. And that might not change unless we change a standard of care. And so we started working on her disorder in the lab. And very early on in our efforts, we also met and connected with an amazing Syngap family who helped support our early work. And since then, we've been pursuing parallel approaches to develop new treatments for STXBP1 and Syngap1 disorders. So the smartest thing, of course, that I did was realize I didn't know what I was doing in this space and to recruit folks wiser than I. And so this is the team that we've assembled called the END team. And I think they're a bunch of rock stars with experts in gene therapy, in CRISPR approaches, in neurogenetics and stem cell technologies. And we really think we need all of this diverse expertise to try and move the field forward on these disorders. So I'm going to skip over the clinical presentation of these disorders as it's been covered or is known to this crowd. And also the role of these proteins, I think, was introduced a bit by Dr. Smith-Hicks and others. And so I'll get right into our approach. So what's our approach to treat these disorders? Here's our 2 copies of either the STXBP1 or the Syngap1 gene, sort of a central dogma schematic. And our kids have a variant in one of these gene copies shown here in red. And depending on where that mutation lies, that can affect any or all downstream aspects of this central dogma. But regardless of the mutation location, a common downstream endpoint is that you're simply not making enough functional STXBP1 or Syngap protein. And so you have this haploinsufficiency condition. And so you can imagine a variety of different therapeutic strategies to address this. You could try to correct the mutant allele directly using gene editing. You can try and add extra copies of the gene with gene replacement therapy, or you can try and boost more out of this normal allele. You could bolster transcription, splicing, translation, protein folding. And from our strategy, we've been focused primarily on manipulating the processing of RNA to try and boost more expression from the wild type remaining allele. And so there are a number of mechanisms that control how much productive RNA is made from a particular gene of interest. And so in general, our strategy is to look for imperfections in this process and then try to hijack or correct those mechanisms to increase the amount of productive mRNA. And so one strategy that we can employ is to try and target alternative splicing, where the same pre-RNA can be spliced into multiple different mRNAs. And sometimes these alternatively spliced RNAs retain certain genomic elements that can target that RNA for degradation, such as a retained intron or a inclusion of a poison exon, which would trigger that RNA for degradation through nonsense mediated decay. So if you can redirect or increase the efficiency of this splicing, you can potentially shuttle more RNA into that productive pool and make more protein. The other element that we've been targeting are microRNAs. So microRNAs are small non-coding RNAs that can bind to an mRNA and target it for degradation or repress its translation. So if we can prevent this microRNA mediated degradation, we could also make more functional protein. So how do we tune RNA biology to our advantage? And this is where antisense oligonucleotides, or ASOs, come in. And these are small modified nucleic acids that can bind to a target sequence on RNA through complementary base pairing and manipulate its biology. And there are multiple ways that an ASO can regulate RNA. The first is to use a silencing ASO, or a gapmer. This is where ASOs with certain chemistries can bind to the mRNA and cause it to become degraded. And this is actually the classic way that ASOs were used, and it's useful in many disorders. Doesn't help us, of course. We're trying to get more expression. The second common goal of ASOs is to modulate splicing, and where you can place an ASO on a specific element, say a splicing enhancer or a splicing silencer, to redirect splicing. For example, to prevent inclusion of one of these poison exons. And these splice switching oligos, these have been used successfully or are being used successfully for the treatment of SMA. They're also looking promising for the treatment of Dravet syndrome. The third way we could use an ASO is to block other RNA binding proteins, such as microRNAs. So in this case, you would put a steric blocking ASO right where that microRNA wants to bind, and preventing that mirror from binding and repressing your target, protecting it from degradation. And so we're pursuing multiple of these sort of splice switching, steric blocking strategies for both STXBP1 and Syngap1. I'm just going to show you one example for each gene today. So this was actually our first fairly simple idea, where if a certain amount of STXBP1 mRNA is normally being degraded due to microRNA engagement, then if we can remove that break with an ASO, we'll have more productive mRNA and more protein expression. And there's really two key questions you have to answer to know whether this could work. One, are there actually any microRNAs in human brains that are normally actively silencing STXBP1? And if so, where are they binding STXBP1, which you need to know to design your ASO to sit there and block it. And so we think this general approach may be broadly applicable to treating other forms of neurodevelopmental disorders, as well as microRNAs silence many genes that are implicated in NDD. And so through transcriptome-wide mapping of microRNA binding sites, the Davidson lab found over about 3,000 genes in human cortex that are actively being silenced by microRNAs with high confidence. And I cross-referenced this list with the 155 best-known genes that cause neurodevelopmental disorders due to monogenic haploinsufficiency. And about half of those are likely under active silencing by MIR in human brains. And that includes both STXBP1 and Syngap1. And so that means for these 70 or so rare diseases, removing that microRNA break could potentially be a useful way to boost gene and protein expression when it's needed. Okay, so can it work? To make a long story short, we've honed in on a few different microRNAs that may silence STXBP1, and this includes this miR-218. And if we take a human neuronal cell line and we treat it with an inhibitor of this microRNA, we can remove that break, and we see an increase in the amount of STXBP1 protein in those cells, which is quantified here on the right. We can also use CRISPR to just cut out that microRNA 218 altogether. And again, we see a similar result with upregulation of STXBP1, also upregulation of SCN1A, which is the genetic basis for Dravet syndrome. We think this is another target of miR-218. So this is a potential opportunity, but this also highlights the problem of inhibiting the microRNA broadly or directly. MicroRNAs regulate hundreds of different target genes, and we don't want to upregulate hundreds of different target genes, which could have unforeseen consequences. We just want to increase STXBP1. So to do that, we need to put our ASO in the right place on STXBP1 to block that microRNA and get specific RNA upregulation. So microRNAs, they can actually bind all over a gene, but they bind most commonly in the 3' untranslated region, the UTR. And this is also where they tend to show their greatest repressive activity. So to determine if microRNAs were binding the 3' UTR of STXBP1, we generated a Luciferase assay with just the 3' UTR of STXBP1, and we express it in these human neuroblastoma cells. And what you see on the right is that only when you transfect these cells with both the UTR of STXBP1 and your antagomer to knock down miR-218 or miR-424, another microRNA we think regulates STXBP1, you see upregulation of the STXBP1 3' UTR. These others are all negative controls. And so this gives us some amount of confidence that these miRs are normally binding and suppressing STXBP1 through its 3' UTR. And so if we just zoom in on that 3' UTR, these arrows denote different potential microRNA binding sites. And if we just zoom in further on this last one, we can look at the putative nucleotide sequence for where microRNA 218 is proposed to bind. And so we first generated about 9 different ASOs, and we put them on 3 of these different potential locations, these miR binding sites, and asked which ones, if any, increase STXBP1 protein expression. And so when you look at the plot on the right here, you see that some of the ASOs targeting this miR-338 site, they did not work. Targeting miR-424 gave a little bit of benefit, and we get a better expression increase with some ASOs targeting miR-218. So this was exciting, but this is just 9 ASOs. We really need to screen hundreds of ASOs to identify optimal leads. And so we're working with Ionis Pharmaceuticals, an antisense company, to try and find the best ASO leads that we are then testing now in iPS neurons that are generated from patients with STXBP1. Okay, so what about Syngap1? I'll share some of a different strategy that we're pursuing on Syngap. So there is considerable alternative splicing on Syngamp, and one element in particular that I think we can take advantage of. And so this is RNA sequencing of Syngamp from adult human cortex. And so this is Syngamp mapped to its genomic locus. So these peaks correspond to exons 9, 10, 11, and 12, and these gaps are the introns. And you see a fairly normal splicing pattern, except for we notice very early on that there's this little plateau down here, right before exon 11. And that corresponds to alternative splicing of exon 11. And within that alternatively spliced region encodes a termination codon, which would then cause transcripts that contain this element to be degraded through nonsense mediated decay. And we've confirmed that it indeed does, as have other groups shown that this leads to NMD. So if we could redirect splicing to the proper start of exon 11, we could potentially get more Syngap and escape this nonsense mediated decay. So we looked for splicing modulators that might be in that surrounding region. And we found that just upstream of that poison exon are a number of highly predicted binding sites for a particular splicing factor that's a well-known regulator of splicing in the brain called PTBP. And so we hypothesized that this splice factor could be promoting the inclusion of this non-productive splicing event in Syngap1. So to test that, we designed a PCR assay to look at that splicing event where we can detect bands at the top here that correspond to the improperly spliced transcript, and at the bottom that correspond to the properly spliced transcript. And if we knock down this splicing factor, PTBP, we see that we switch the ratio. This is data in hex cells and in Shushi cells. And we can indeed get more of this bottom productive band while reducing this top non-productive band. Now, importantly, corresponding to that correction of splicing, we get an increase in the amount of Syngamp productive mRNA when we deplete this PTBP. So just like with the microRNA approach, though, we don't want to just inhibit PTB broadly. And we've done splicing analysis of what happens when you do this, and it's regulating over 1,000 different genes in human neurons. So we want to specifically block its interaction with PTBP. And again, this is where ASOs come in. And we put many different ASOs on several different predicted or measured PTBP binding sites on Syngap. And so sparing the gory details, we've tried about 100 different ASOs to do this. And these are some of our best results. So with a particular ASO, for example, this is this SSO19, we see that when we treat cells with higher and higher concentrations, we increase the amount of proper productive splicing, and we increase the amount of Syngap gene expression. This is our current favorite sort of home run ASO right now, ASO 85, where you see this really robust induction of Syngap mRNA expression over 5-fold. And this does indeed translate to a multifold increase in protein expression in these cells as well. So this gets us very excited. If we could increase protein expression 2-fold in a patient's brain cells, that could correct the underlying deficiency. But it's really important to note that these are not patient brain cells, right? This is a screening cell line, and this is quite some distance away from a child's brain. So how do we move this forward? How do we get a little closer? We can't take brain cells from our kids, but we can take a blood draw, and we can use induced pluripotent stem cell technology to turn those blood cells into stem cells and differentiate those stem cells into neurons that harbor the genetic architecture of our patients. This is what those neurons look like. These are neurons from a patient with one of these two neurodevelopmental disorders, and we can measure the neuronal activity, their synaptic function, and when they don't have enough STXBP1 or Syngap1, and these patterns are disrupted. So it's sort of where we are. We've generated iPSCs from both STXBP1 and Syngap1 patients. Here's the example from Syngap, where this patient had a nonsense variant that led to the introduction of a premature stop codon, this R1240X mutation. And we've confirmed that indeed this leads to a clean haploinsufficiency that's reproduced in these patient cells. You see a reduction in the Syngap mRNA levels by about 50%, which corresponds to that mutant allele being robustly silenced by nonsense-mediated decay. And as you would expect then, you see this similar reduction in Syngap protein expression. And we can use CRISPR tools to actually correct that underlying mutation. And we see that that restores the levels of both the mRNA and protein, sort of confirming both the causative nature of that variant and giving us this nice isogenic pair of cells for testing. So this is where we are. We have another patient line also about ready for testing, and we're treating these with our different ASOs to try and find the one that restores the most amount of protein and the most amount of function in these neurons. And that's what we'll try and push forward into a first-in-human application. So a quick thanks to all the amazing folks who did this work, and really all of these individuals contributed to the different aspects of the work I shared today. It's a very collaborative team, and that's Lucy thanking you for your attention as well, and to our funders who are listed down here below. I'm happy to take any questions if there are still some time.
Thanks, Ben. Shameless plug, one of your collaborators, Liz Heller, is a Syngap and an SRF grantee. Because I want to thank all of our families who donate and point out how critical it is to support scientists like yourself to do this work.
This is an awesome colleague, and we're very excited about her line of work as well, which we didn't have time to talk about today.
But it all helps.
There is one question in the chat. Is there an ETA to getting to human trials?
The answer is not a precise one. Part of the reason why we chose to pursue an ASO route early on when we were thinking about these different strategies is that we thought it gave us our most rapid chance of getting to the clinic, the fastest possible route. And there is a demonstration, of course, a rather famous demonstration of an ASO being developed and given to a child in need, in dire need, within about a 1.5 year timeline. And so given the relatively good safety profile of ASOs and their other approved clinical use, the drugs that you're working with in your in vitro studies are the same ones that can go into the patient. And so there is the possibility of rapid translation without going through extensive testing, perhaps in large animal models, for example, which would still have to be done for any sort of CRISPR or some of the other genetic approaches that are pursued. So there's a possibility of a quick timeline, but we can't nail that down precisely today until we have an important meeting with the FDA. I see a separate question about who's funding Dr. Prosser's work. I'm not sure if that's something that I should answer. The NIH, and we have a sponsored research agreement with Ionis Pharmaceuticals, who are both supporting our work. I'll leave it at that. We've also benefited from philanthropic donations. Thank you all very much.
Thank you, Ben.
Yeah.
We're all rooting for you, and I'm sorry your child is sick, like ours are sick, but we are so grateful that you're you have the talent that you have, and you're part of the team that you're on, and that you're working. So
should I answer this last question that just popped up, or
yeah, go for it.
Okay. What is known about delivery of ASO into cells in the brain? How similar is the set of cells each time an ASO is delivered? That's a really great question. There's still a lot of mysteries about how ASOs get into cells and their biodistribution. There is some mechanisms that are understood. I think how those mechanisms vary between different cell types in the brain is not well understood. The delivery method is currently being done through intrathecal injection, so into a spinal tap, which is not really a pleasant experience, of course. And this has to be repeated, say once every 3 months, 6 months, depending on the lifetime of that ASO. There are additional delivery routes that are being pursued now in terms of implantable devices that could have slow release technology. So it would be a one-time procedure for a child, but I don't believe those have been clinically approved yet. In terms of the efficacy, though, however, the work, particularly with spinal muscle atrophy and Spinraza or nusinersen, gives us a lot of confidence that this can be an effective therapy to consistently regulate alternative splicing in the brain or in the central nervous system.
Great. And is one more question in the chat. Have you guys had a pre-IND meeting with the FDA, and did dose finding come up?
No. We're waiting for some additional data in the patient neurons to have that meeting.
Well, thank you so much, Ben. It has been a wonderful roundtable. I'm truly appreciative of all of our researcher collaborators. And Geraldine, I'll let you close it out.
Well, a big thank you to all of our presenters and all of the attendees just for taking the time today to learn about synaptopathies and to learn about the approaches that are being used in the synaptopathies. Hope for those of you who are epileptologists, you work primarily with patients who have epilepsy. I hope you'll remember to think about synaptic causes of epilepsy when you're meeting new patients. Thanks again. And Mike, thank you so much for organizing this event. We really appreciate all your work and efforts.
Yes, thank you.
Okay, guys, have a good one.
Take care.